Hello students, this is Geethanjali. In the previous video we did replication and in this video we'll be doing transcription. So replication is the process in which the DNA makes another copy itself, right? So transcription is a process in which the RNA is formed from DNA. Okay, so it is basically the process of copying the genetic information from DNA to RNA. So in this, only one strand of DNA is involved. That means when we have two strands of DNA like this, only one strand of RNA is formed using one of the strands of DNA. Why is that? So this is formed again by using complementarity. So the only difference here is that here we will have uracil instead of thymine. So wherever there is A in the DNA, the complementary base will be U in the mRNA, that is the uracil and not thymine. So let us understand why only one strand of DNA gets transcribed. So first of all, complementarity does not mean the same. So these two strands, they are complementary to each other, but when they are transcribed, the proteins that we get from them, they are not the same. They are different proteins. So that does not really help. And on top of that, whenever both of the strands get transcribed, we get two RNAs. And these two RNAs will be obviously complementary of each other because these were complementary to each other before, right? So they will form a double-stranded structure. And this double-stranded RNA will not help us in getting translated to proteins. So that is why only one of the DNA possibly gets transcribed. So to understand transcription, we need to understand the transcription unit. So a transcription unit contains three things. One is the promoter part of the this is basically the double-stranded DNA from which is going to get transcribed. So in this DNA we have a promoter site, we have a structural gene, and we have the terminator site. So we have three parts to this transcriptional unit. Like in replication, there were many enzymes. Here we have an enzyme called the DNA-dependent RNA polymerase. And just like DNA polymerase, this too transcribes or adds nucleotides from 5' to 3' direction. That is, it can only add to 3' end. So because of this, as it runs from 5' to 3', it forms the RNA on the DNA strand which runs opposite, that is 3' to 5'. So this will be our template strand. That means using this strand, the RNA polymerase will put the corresponding nucleotides and make the RNA. So in this case, you can see this strand will be our template strand because it runs the other way around. And that is why we call this template strand too, because we use the strand to form the RNA. And you can see the other strand. You can see the coding strand. Another strand. This is the non-template strand, or we can also call this coding strand. So why did they call this the coding strand when this actually does not code for anything? Because we know that the template strand is used to form the RNA. So why is this called the coding strand? And this is called the non-coding strand. So why was that meaning? Simply because when the corresponding RNA is formed, let us say we had a sequence like ATGT was transcribed, so the corresponding RNA sequence will be UACA, right? Now, if you tell me if this was the template strand and this was the RNA transcribed, the what would be the corresponding code of the coding strand? It will be, you can see how similar the transcribed RNA and the non-template strand are, right? So that is why they probably named it coding strand, because we can identify the code of the RNA just by looking at our non-template strand and just by replacing the T with U's. And also, one thing about the location of promoter and terminator gene is that we say that the promoter gene is present in the part, that is the 5' end of our coding strand, and the terminator, it is present in the downstream part, that is the 3' end of our coding strand. Again, here we take the reference of coding strand because it is easier for understand, because we usually consider 5' as the end from where the nucleotides are added, right? So that is why we call this upstream and this here downstream, and we take the reference of coding strand here, that is of this 5' and 3' of this coding strand. So before going to the actual process of transcription in prokaryotes, let us learn few terms. First of all, what is cistron? So we know that gene is any DNA sequence that codes for biomolecules like either nucleic acids like RNA, etc., or even proteins, right? But cistron in particular is the DNA sequence that only and only codes for proteins, or you can say polypeptides. In this, we have two divisions, okay? Monocistronic and polycistronic. So, monocistronic means this particular DNA sequence, this cistron, codes for only one polypeptide. Whereas polycistronic means that particular DNA sequence codes for many polypeptides. Prokaryotes, that is bacteria, are polycistronic, whereas eukaryotes are monocistronic. When talking about monocistronic, two other terms come into play: exons and introns. So, exons are that part of the DNA sequence that are functional and code for amino acids that make up a protein, whereas introns are those DNA sequence that come in between the exons. Okay, these are like interruptions in between that actually do not code for any amino acids, and thus they are not transcribed. So these are called introns. So the expressed sequence, or we can say the one that actually codes, or the coding sequences, are exons, and these exons are interrupted with introns. So these are those sequences that are not required in the RNA, which will later be translated into proteins. Coming to the actual process of transcription in prokaryotes. So, in general, there are three types of RNAs. Okay, we have mRNA, we have tRNA, and we have rRNA. So, this is basically the transfer RNA, this is the messenger RNA, and this is the ribosomal RNA. Now, the kind of RNA that we are going to form with the help of transcription are all these three. Okay, but the type of RNA that helps us in translation is mRNA. Okay, this mRNA basically has the sequences, the coding sequences that correspond to amino acids, whereas tRNA is that RNA that will basically read this a particular sequence and then form the polypeptide chain by using amino acids. And whereas rRNA also helps in the protein synthesis, basically they're present in ribosomes, so we know that ribosomes play a part in protein synthesis. So there are three basic steps: initiation, elongation, and termination. Whereas elongation is completely taken care of our DNA-dependent RNA polymerase, initiation and termination require a factor. Okay, so initiation requires initiation factor, whereas termination requires termination factor. So the initiation factor over here is the sigma factor. So this sigma factor helps to find the coding sequence to start off with. So first it attaches itself with the RNA polymerase, and that's when the initiation begins. The RNA polymerase, again, it uses nucleoside triphosphates as substrate, and when elongation happens, the sigma factor is again removed, and the RNA polymerase alone catalyzes elongation, and it keeps adding nucleotides corresponding to the sequence of the template strand. And then finally, to terminate this process, another factor called the rho factor is required. So this rho factor is the termination factor, and it helps in terminating this process. Now, as the RNA is being formed, as I told you, the sequence here is polycistronic, and unlike in eukaryotes, it is not interrupted with introns. So we can directly sequence this entire RNA into proteins. So translation can simultaneously be happening as the RNA is being formed. In case this is mRNA, so simultaneously the translation happens in the prokaryotes. Coming to the same process of transcription, but now in eukaryotes, it is a bit complex because of the presence of introns, etc. So it needs a bit processing. So as we saw in prokaryotes, there was only one type of RNA polymerase, whereas in eukaryotes, you will find three types of polymerase. So we know that ribosomes have two subunits, right? In eukaryotes, we have 60 subunit. This is the bigger subunit, and we have 40S subunit, that is the smaller one. So the 60 subunit, it contains 28 sRNA and 5.8S And whereas our 40 subunit contains 18S rRNA. So the RNA polymerase 1 is responsible for the production of 28S, 18S, and 5.8S RNA. All these, right? Whereas RNA polymerase 3 is responsible for the formation of tRNA, 5S RNA, and snRNAs. So snRNAs, they play a role in processing of the RNA, which contains some sequences from the introns. So we'll see that later. And RNA polymerase 2, it is responsible for the formation of hnRNA, that is heterogeneous nuclear RNA. So this heterogeneous nuclear RNA is nothing but the precursor of the actual mRNA. As I told you, we have introns also, so we need to get rid of introns. So this hnRNA basically contains the intron sequences, and we need to get rid of them to get the actual mRNA. Now, when RNA polymerase 2 forms the hnRNA, it needs to undergo processing before getting translated. So we have these three steps here, that is splicing, capping, and tailing. So splicing is the process where the introns are removed. So you can see we have introns over here, and you can see here these are basically SNRPs, okay, or you can also call them snRNAs. So these basically snRNAs help in the removal of the introns. Then we have capping. Capping basically involves the addition of a nucleotide at the 5' end. This nucleotide which is attached, it is the methyl guanosine triphosphate. So this addition of this unusual nucleotide to the 5' end of the hnRNA is called capping. Another process, that is tailing, or we can also refer to this as polyadenylation. In this process, some 200 to 300 adenylate residues are added at the 3' end of the hnRNA. So this completes the formation of our mRNA. So at the end, we finally get our mRNA, which is ready to get translated and form polypeptides. You see here that this splicing, okay, where the introns are removed, is a very essential process, because we don't need these introns to get translated, because they do not code for anything, for any amino acids or anything. So, and this can only be done with the help of our RNA, that is the SNRPs, snRNA. So we say that this process of RNA actually shows the dominance of RNA world. So this was about transcription.